So today we have the honor of introducing Dr. Jack Gallant, a distinguished neurophysiologist at UC Berkeley with a long history of working on primate vision. Dr. Gallant received his PhD from Yale University and completed his postdoctoral work at Caltech and Washington University School of Medicine. Today, Dr. Gallant is Chancellor's Professor and Class of 1940 Chair at UC Berkeley, with affiliations spanning several disciplines, including psychology, electrical engineering, computer science, bioengineering, biophysics, neuroscience, and vision science. Not only is Dr. Gallant preeminent in his field for research, but he also remains steadfast about educating the public on neuroethics through his post at the Colby Center for Ethics, Science, and the Public. He tackles issues such as the implications on privacy, autonomy, and agency in a world full of fast-developing neurotechnology. He regularly gives presentations on the implications of neurotechnology and the law to circuit courts, the National Academy of Sciences, and was even featured on the Freakonomics podcast.
It was a really good podcast. They did a great job.
Dr. Gallant is perhaps most famous for developing the most accurate brain decoding models for transforming fMRI data into visualizations of what a participant is shown in the scanner. This discovery was named one of Time Magazine's Inventions of the Year in 2011. With over 20,000 citations, 40 plus published papers, and millions of dollars in NIH funding, and an H-index of 57, Dr. Gallant's work has greatly enhanced the field's understanding of the structure and function of the visual system. These days, Dr. Gallant and his lab focus on developing models for how the brain encodes information during naturalistic tasks, incorporating both the intrinsic and extrinsic experiences of the world. The Center for Mind, Brain, and Culture and Frontiers in Neuroscience have invited him here today to discuss the development of distributed conceptual network theory as a scaffold for consciousness and working memory. We've been eagerly awaiting his talk, so without further ado, please join us in welcoming Dr. Gallant.
Hi, well, thank you very much for inviting me. This is a very diverse group, a very diverse audience of academics, and I wasn't really sure what the division would be between neuroscience and society and culture. So I have kind of two talks I've been giving lately on two projects in the lab that are both long-term projects. One is on the representation, sort of what we call the Distributed Conceptual Network, which is the representation of stuff in the world, and how it interacts with working memory and attention to sort of give you a scaffold for daily life and ongoing experience. And the other project is on navigation in virtual reality. And I decided to mostly focus on the conceptual network stuff, although I'm going to go into navigation in one part of the talk for reasons that will become obvious when we do that. I always like to begin with just an example that's kind of motivational. This is a brain, human brain. It's inconveniently folded up to be as small as possible. So to analyze it, we computationally inflate it, and then we flatten it out like a pizza. In fact, if we did something like that to your brain, we would have something about the size of a large pizza. And in this particular map, the visual system is in the middle here, which is the back of the brain. Prefrontal cortex is here and here at the far left and far right, and the motor and somatosensory cortex are kind of in the middle. And auditory cortex is in the middle at the bottom. Now, you can see here that the person's just watching a movie. There actually happens to be sound with this movie, but it doesn't really matter. This was an open source art movie, actually from a film school student, and it's actually the story of life, but that's not really important. You kind of figure that out about halfway through the movie. Now, the question there is, what happens? Oh, I didn't describe the bottom. On the bottom of this slide, we're measuring metabolic brain activity as measured using functional magnetic resonance imaging. And the red locations on the map at the bottom indicate areas where, during that particular point in time, brain activity is higher than it is on average. And the blue locations are locations where the brain activity is lower than it is on average. So if you watch this map evolve over time as humans watch this movie, you can see that the pattern of brain activity changes dynamically. There's always something new happens, some new configuration of brain activity, and this reflects all of the experiences people have while they're watching this movie. Certain parts of the brain, the visual parts of the brain at the back of the head in the middle of the slide, are going to represent the structure of the visual scenes. There's auditory activity here because there is sound being played in this movie, although you can't hear it. There are parts at the prefrontal cortex you can see is highly activated when the movie becomes sort of more abstract, when there are religious themes or themes involving social interactions or human relationships. You see a lot of activity in prefrontal cortex. Kissing turns out to elicit the largest amount of activity. Students are very interested in this. You can see that the patterns of brain activity are rich, right? And so it makes sense because anything you can think about or experience or be associated to this movie must be reflected in brain activity, right? We're not dualists in neuroscience. We'll believe that the brain gives rise to all experience. Therefore, any experience you can have must be measurable in the brain somehow, somewhere. And with MRI, we just have a kind of a low resolution measurement of the brain. And my job as a cognitive neuroscientist and a systems and cognitive neuroscientist is to try to figure out how different regions of the brain represent information in this movie or information that's associated with this movie. And since I like impossible challenges and I'm kind of a megalomaniac, my view on my job is I want to be able to perfectly predict how every single one of the 100,000 points I'm measuring across the surface of the cortex responds to whatever they're responding to in or associated with this movie at all points in time. And when I do that, then I'm allowed to retire. So I'm getting old, and I'm trying to get as much done as I can before that happens. All right. Now we're measuring, all the measurements I'm going to talk about today are from functional MRI, which my joke name for functional MRI is functional hemophrenology. We're not measuring neurons in MRI, we're measuring metabolism. But I just want to tell you, for those of you who are like neurophysiologists and wonder how MRI works, or maybe you are just never seen an MRI machine before, just want to give you 2 quick slides on MRI. MRI is basically a chemistry experiment. It's a bulk chemistry experiment, and in this particular device, your head is the sample. And we put your head in a strong magnetic field, and it aligns some vanishingly small fraction of the protons along the main magnetic field. Then we put in some radio frequency energy at different angles and different phases and different frequencies, and this makes the protons spin off axis. Then when we turn off this gradient of energy that we're supplying to the brain, the protons spin back down to be aligned again with the main magnetic field. And when they do that, they give off energy that we can measure with an antenna around your head. And since this is a 3-dimensional spin, we can measure it with 2 degrees of freedom. And we can look at the rate that these spins dephase over time. And these rates are related to the kind of tissue. Basically, protons that are in water, like the ventricles, will dephase at a certain rate. Protons that are in fat, like white matter, will dephase at a different rate. Protons in gray matter will dephase at a different rate. And most importantly, for today's talk, protons dephase at different rates if the hemoglobin is oxygenated versus deoxygenated. So this, if you tune it correctly, can be used as a measurement device that measures the rate of oxygen change in the local area of cortex. And we're talking about a size of an area about, say, at this resolution, 2 by 2 by 2 millimeters. Right. Now, oxygen is important in the brain because neurons are little chemical engines that extract sugar and oxygen from the bloodstream to make ATP, and ATP is the fuel that runs all neural processes. And so evolution, being really cool and really weird, has developed this very strange second messenger system for regulating blood flow when neurons fire. Basically, there's some neurotransmitter, it crosses the synaptic cleft. The extra neurotransmitter you don't want to just leave in here because that would cause paralysis. So you have astrocytes that soak up extra neurotransmitter, repackage it, and deliver it back to the presynaptic neurons. And as a side effect, these astrocytes have little philopodia that surround the local blood vessels. And when the astrocyte goes to work sucking up neurotransmitter, it also releases its philopodia on the local blood vessels, thus increasing the size of the blood vessel, which increases the blood flow, the blood volume, and changes the local blood oxygenation. And that's what we measure. Okay, it's really weird, and you might think this would be impossible for this thing to even work. But the amazing thing about MRI is actually this works pretty well. And really, this measurement, is the same measurement if you go back to like old, well, people still do this, but old like intrinsic optical imaging studies from Grünwald and other people in the 80s and 90s, still used a bit today. This is exactly the same mechanism that they were looking at. So this is a real biology. It's just at a low frequency, okay? So now I have, a low spatial resolution. So now I have my x variables, which are whatever my task is. In this case, the movie I just showed you. It might be listening to a story. It might be watching a movie. It might be a person in virtual reality. And then I have my y variables, which are my brain measurements. And I want to relate my x's to my y's. And that, like most of science, is standard regression problem. So if we want to solve this as a standard regression problem, how do we do it? Well, the one thing we know how to do really well in science is linear regression. So what we would like to do is turn this really hideously complicated problem into a linear regression problem. And there are good ways to do this. The general linear model is a very good model. We know all the theorems involving the GLM are provable. We know exactly when it will work. We know when it won't work. We know what its assumptions are. So what we can do is we can turn our problem into a GLM problem. And the way we do that is we define feature spaces that extract information from the stimulus and task nonlinearly, and then we use regression to relate those features to the brain. To make it concrete, imagine you're listening to a story, and I want to know if there's some part of the brain that represents the meaning of the story. So I might use latent semantic analysis or a deep neural network like BERT to extract the semantic meaning of the stories and translate the stories from the original sound spectrogram into an embedding that reflects the meaning of the stories. And I might have, say, 2,000, 5,000, 10,000 meaning features, where each one of those features says that this part of the story, relates to a certain semantic concept like a dog or a building or love, right? And then I would just simply take this feature vector and regress it onto our brain activity. So that's really all we're doing here. This is called an NL model, nonlinear linear model. And you can always solve any problem this way because you can stick a linear system at the end of any arbitrary system, and all it does is rotate the coordinates and it doesn't change anything. So as long as the system isn't changing over time, we assume it's stationary, it's not learning, then this will work. So we take our pixels by stimulus matrix, we extract features. There might be, say, semantic features, sound spectrogram features, maybe some other features. Now we have a bunch of features by time matrices. We stack them all together. We basically do regression, and we get basically a set of beta weights. So for each of the 100,000 locations in the brain that we're measuring, we're going to end up with a set of beta weights that are a regression equation that tells us how those specific features that we regressed are represented in that specific voxel. And there's all software to do this that's public that I'll talk about at the end if you guys are interested. Okay, so the advantages of this approach, this is called an encoding model, and because we're doing it in voxels, it's called a voxel-wise encoding models or voxel-wise modeling. The advantage is that all the assumptions about the models and hemodynamic coupling are completely explicit in this framework. There's nothing hidden. There's no operational definitions. It's an explicit definition in terms of a program. Permits the use of complicated natural stimuli. We can test multiple hypotheses. In one of the experiments I'll show you later, we test 35 different feature spaces simultaneously. It maximizes the information we can recover from the brain. It gives us functional maps in each individual participant, not just at the group level. It gives us statistical significance, which is the noise floor, right? Statistical significance is just non-randomness, just means my data is not completely random. But it also gives us prediction accuracy, effect size, and generalization tests. And all of this stuff is implemented in software that is freely available from our lab. The disadvantage is, if we're going to fit a high-dimensional regression model to each one of these 100,000 voxels in the brain, we're going to need really good data, which means we have to be very careful about how we do the MRI experiment, and we have to collect a lot more MRI data than you would in a typical experiment where you, say, only have two conditions. And also, I'm not maximizing power, statistical power, with respect to any hypothesis, because it's a very data-driven kind of approach. There's no free lunch in science. There's always costs and benefits. Okay, so to show you how this works in this domain of, domain of conceptual understanding, I'm going to focus on a series of experiments that looked at this sort of dynamic conceptual network. And a lot of people have been interested in this, how you represent conceptual information. For hundreds of years, philosophers and psychologists have studied this problem. Currently in psychology, there's two main theories. Well, I should say there's one main theory and a side theory that nobody pays attention to. Okay? The main theory is this hub and spoke theory, most recently promoted a lot by the Lamp and Ralph group, that says basically you have and sort of amodal or multimodal conceptual hub that represents all kinds of different conceptual information, and it's located in the anterior temporal lobes. And this is linked to a bunch of mode-specific conceptual representations, like just for vision, or just for audition, or just for olfaction. And they all come together in the anterior temporal lobe. Now you might wonder did this model evolve? And it evolved because if you give a human anterior temporal lobe damage, either by a stroke or more often by anterior temporal lobe dementia, they lose their semantic knowledge. They can navigate through the world, but they can't really talk about anything. They don't really understand how the world works. They can't make predictions. Their general conceptual knowledge of the world just completely degrades. And they tend to become monomaniacal and just like obsessive, focus on one thing, and it's oftentimes socially inappropriate. One of the patients that we study goes out in the backyard every day and then burns ants with magnifying glass and then eats them. So that's obviously not normal behavior, right? So the ATL is clearly doing something very, very important in conceptual information processing. There's another theory of the conceptual information that has evolved over time from Jeff Binder's group at Medical College of Wisconsin. And he was really the first person that kind of pushed this forward, and I don't think he gets enough credit for it. His view was based on he's a neurologist, and based on looking at a lot of brain lesion information, he came up with the idea that this conceptual information is actually broadly distributed across a network of areas. The ATL just happens to be maybe a bottleneck route to memory. Because of course, your ongoing conceptual experience, your experience of the current world, involves both input from the world and input from memory. And those two things have to be combined in what psychologists call the internal workspace, or the blackboard, or the stage, right? Your ongoing experience. Okay, so I'm going to argue in this talk that based on a lot of data from our lab, that there are really 4 components of this network. There's an amodal, or if you're really neurotic, multimodal conceptual network that's broadly distributed across the brain. There's multiple modal conceptual networks, and then there's a long-term memory access that goes through the ATL, and then there's executive control of these amodal conceptual representations. And I'm going to go through the evidence really quickly for these networks. This is data from an experiment where people watched movies. These movies do not have a narrative arc. They're clips that are 10 to 20 seconds long that are all shuffled up together. So there's no long-term narrative information. And in this particular case, you find that there are a lot of locations in the brain that represent semantic information in the movie, meaning the categories of objects and actions in the movies. I don't have time to go through this graph in detail, but you can look at the very old, at this point, paper if you wish. The colors here just show you how different kinds of conceptual information represent across the cortex. And this broad band you can see here that's sort of vertical next to, remember in this map, as in all the maps, the visual system is in the middle and the prefrontal cortex is at the far left and far right. So you can see these broad vertical bands bracketing the middle, and these are the semantically selective visual areas that are essentially form a concentric ring around the retinotopically organized visual areas. So we know there are many regions of brain that represent conceptual information in movies. If you have somebody listen to stories, just narrative stories, and then you map the representation of lexical semantic concepts in the stories, you find that the lexical semantic representations are also broadly distributed. In this map, again, these colors correspond to different kinds of lexical semantic concepts, and the colors correspond to the legend down at the lower right. So green regions on this map are regions of the brain that tend to represent visual concepts. Red regions are regions that represent social concepts. Purple regions are regions that represent mental concepts, and so on. In fact, in the vision experiment, we mapped 1,700 distinct semantic concepts, and in the story experiment, we were mapping about 950. So there's a lot of different concepts that are represented in this map. We're just showing you a kind of a low-dimensional projection here to try to give you an overview of the information that's represented.
Hey, Jack.
Yes?
Is there a big overlap between your visual and lexical semantic concepts? If you put one brain on top of the other with these, is there a big overlap, or you're trying to really make a distinction?
That's a really ugly question, and for a reason that I will get to later on in the talk.
Okay,
but I'll give you the preface now. These maps are not static. these maps are heavily modulated by attention.
Okay.
And the trouble we've had in answering this really, really critical question is making attention exactly the same between a story and a movie turns out to be way harder than you would think.
Fair enough.
And so generally they're associated, but there are substantial differences, and we haven't been able to determine if those are differences due to the underlying scaffold or due to attention.
Yep.
It's an open issue at this point.
I'm only asking because you had said something about amodal, or if I want to be picky, supramodal, and I don't know how you make that if you're not saying that there's an overlap between perhaps vision and
Perfectly valid point.
And
so
lexical semantic.
I'm glad to see
But
I'm glad to see you're paying attention. So I say amodal because I'm basically lazy, and it's kind of, it's vision, and it's responding to vision, it's responding to language. That's kind of amodal to me. But the more neurotic people who are better scientists than I am, who work in this field, say no. To show that it's amodal, you have to show A, that it's multiple modalities, and B, that it's the same thing being represented across those modalities, and we have not done that yet.
Okay.
So a lot of these slides might say amodal because it was being sloppy, but the one valuable thing about grant reviews is they definitely teach you how not to be sloppy. So I'm reorienting my language in this because I think that criticism is valid, right? We have shown that, for example, in this region of prefrontal cortex, there's language and vision stuff both being represented, but we haven't shown that the representations are the same.
Okay.
So therefore, we can't really say this is an amodal representation of a concept. We can only say it's multimodal representations of conceptual stuff,
Good, yeah.
right? And thank you for asking that question. Okay, so one thing you might wonder is, well, wait, I can read a story or I can listen to a story. Is that going to lead to different kinds of representations? The answer is no. You can see this conceptual map you get from listening. It's exactly the same as the conceptual map you get from reading, except for the differences in visual cortex and auditory cortex, right? One of them is stimulating higher order auditory cortex, and one of them is stimulating visual cortex. But other than that, these maps are basically the same. There's an online brain viewer. If you guys, a lot of people have seen this brain viewer by now. If you haven't seen this brain viewer, I suggest you go look at it. It will provide at least 5 minutes of pure neuroscience joy and entertainment. It allows you to interact with the data from our stories experiment, where you can click on individual voxels and then find out what they're tuned for. So this shows you that this voxel, in fact, all these red voxels I'm clicking on are tuned for social kinds of concepts. Family, mother, father, brother, and then bad things that happen to families, usually like divorce, death, cheating, things like that. Then in a second, you're going to see I'm going to switch and start clicking on these dark green locations. And these dark green locations are regions that are a number network. And these represent numbers, time, units of measure, like ounces and gallons and miles, and money. All those things tend to be represented together in a number network. And in fact, for basically any kind of family of concepts you can think of, there's a whole network of brain areas that represent these concepts. And that's true for both families of concepts like social things or vision things or number things. It's also true for individual concepts. So for example, you could just map dog, where is dog represented? And you can see that all these red locations in the brain are places that are predicted based on these models to respond when you have a story about a dog. And then the question is, well, why are there so many different locations that represent information about dogs? I have a theory about this, which is I just pulled out of an orifice. I don't, there's no data supporting this, but I like it. It's a good sounding theory, which is I think that these different spots that are all kind of dog selective are representing different aspects of dogness. So for example, the dog selective regions near the visual system are representing how dogs look. Dog selective regions near auditory system are representing how dog sounds. Dog selective regions near, say, olfaction might represent how a dog smells. And dog selective regions in prefrontal cortex are interacting with your memory system, and maybe you remember that you were bitten by a dog when you were young and you don't like dogs, right? All that information in conscious experience comes together in some unitary whole by some magic that we do not understand, and I will predict we will not understand it, certainly before I'm dead, and I suspect not before you're dead. All right. Now, one of the things people like to do in cognitive neuroscience, because the great thing about these MRI experiments is you see the whole brain. You see everything. You see these networks of stuff at low resolution, like, a plane flying across the continent of the United States, right? And so it makes you think, well, wait, where are the networks? I would like to put networks together. And so to do that, we have developed a method called modal connectivity. Modal connectivity is basically an alternative to functional connectivity. And for those of you who don't do functional connectivity, I'm just going to tell you quickly, functional connectivity is simply cluster analysis done on the correlation matrix, where you're looking at the correlations between voxels in an experiment. So the experiment might be resting state, in which case you're not controlling all of the variables controlling brain activity or latent variables, or it might be a task, or it might be listening to stories. But there's some conditions under which you're recording voxel activity. So this is a time series of bold signals, and then you correlate them together, and you end up with, and then you do cluster analysis, and you end up with something called functional connectivity, and you can make that into a graph if you wish. The problem with functional connectivity is it's all based on correlation, and correlation is a really bad statistical model, because correlation assumes that the two variables, that the noise in your two variables is independent and Gaussian, and that there's a linear relationship between these two variables. And one of the things we know in MRI data is your noise is nowhere near Gaussian. It is not Gaussian. In fact, the way we pre-process MRI data, the way everybody pre-processes MRI data, is they take all the noise that's not Gaussian and they try to remove it and hopefully just keep the Gaussian stuff left. MRI data are hugely biased by non-Gaussian signals that have to do with MRI physics and susceptibility artifacts having to do with different tissue interfaces and weirdnesses having to do with MRI physics and the magnet that nobody in neuroscience cares about. And those kinds of contaminations leak into functional connectivity data and cannot be removed. So after spending about 10 years trying to come up with a better way to get functional networks than functional connectivity, one of my postdocs, Matteo, came up with the brilliant solution of doing it the stupid way. The stupid way is, okay, what do I have here? What's my data? I've got 100,000 voxels. And each voxel is a vector of regression weights. Say it's a 1,000 long vector. So I've got a 1,000 long vector, 100,000 voxels. Let's just cluster those vectors. Turns out that works great. Why didn't we do that 10 years ago? It's fantastic. There's a bunch of data science things you have to do to cross-validate it and make sure you're not overfitting and make sure it's optimized. But the method itself works really, really well. So we're going to take these semantic vectors for our voxels in the story listening experiment, and we're just going to do cluster analysis on them and pull out functional networks. To see how many functional networks to pull out, we just cross-validate across individual people. So you can see that after you pull out about 13, this is a hierarchical cluster analysis, right? So we can pull out as many different kinds of clusters as we want. 13 networks gets you most of the way there. The optimal answer is actually about 135 networks, but we only have limited time here. If I told you about 135 networks.
Optimal answer to what?
What?
Optimal answer to what? Sorry, I lost.
This is optimal predictions out of set. So
Okay,
basically you take a
got it.
subset, you take a subset of the subjects, you find the clusters, and now you're going to predict
These are left out.
voxel activity in left out subjects. And then you average over all the left out subjects, and that's the number,
Okay.
right? So we're using prediction accuracy as our metric of which networks to pick. You should come to all my talks. You ask great questions. Anything that's not clear, just feel free to chime in. All right. So obviously this is insane. I'm not going to tell you about this. So let's just focus on these 13 networks here. Okay, so this just shows you the 13 networks. This is 13 networks that you get from the best model connectivity solution. And all these gray things are where there's just noise and you can't fit a network. And this is the 10 networks you get out of functional connectivity. You can see a bunch of these networks are just fitting noise, and they're much broader, and they don't really respect the boundaries of this model. So for a lot of reasons, you can see the bioarchive paper if you're interested. This method we think works way better than functional connectivity. So let's look at these networks. Well, okay, so here's 3 networks that all represent physical things. You can see these are word clouds that represent essentially the concepts at the centroid of these networks. So this is material properties of objects. This is actions that can be performed. This is measurements that can be taken. There's place networks, traveling locations, indoor locations and outdoor locations. There are a variety of social networks, social visits, communication, written communication, relationships. There's discourse, joking and arguing. Turns out to be 2 separate networks. Oops. And this is just blow-ups of these slides I just showed you. So there are a lot of different networks, and each of these networks has a different pattern of, essentially modal connectivity across the cortex, right? Now, I assume if I have 2 random voxels in the brain, and they have 1,000 long semantic tuning vectors, and those vectors are highly correlated with one another so that they go in the same cluster, I cannot but assume that there's some underlying anatomical connectivity that's giving rise to that, because that's very unlikely to occur by chance. Skip this. All right, now one question we've also pursued is the relationship between the visual modal networks and these multimodal networks. So to do that, we compared the story semantics features to the short clip semantics. I just want to be clear what's going on here. Story semantics is basically semantic features we're fitting to voxels in an experiment where people just listen to stories. These are moth stories that Hassan first started using. They're great. People love listening to moth stories. And this short clip semantics is semantic models we're fitting the visual system when people are watching short clips of movies that are all scrambled, so there's no narrative arc. Okay? If there's a narrative arc, these two networks end up overlapping. But if there's no narration, the vision network is basically separated from the story semantics network. You can see that there are a lot of blue voxels, a lot of red voxels, but very, very few white voxels. And any voxel that represented semantic information in these short clips and stories would be white. So these two networks are kind of divorced from one another. If you don't have narrative in vision, you activate a bunch of visual information. If you have narrative in stories, you're activating the narrative network, right? These are two different kinds of subnetworks within this larger conceptual network. And the cool thing is if you look along this border here, outside visual cortex, if you just focus on the middle of this slide, you see that there's a bunch of retinotopic visual areas that are red. And immediately anterior to those retinotopic visual areas are a bunch of areas that respond to stories that are shown in blue. So these high-level red areas are responding to the semantic content of movies, like a dog in a movie or a truck in a movie, but they're not responding to stories. And the blue areas immediately anterior to them are responding to, say, a dog in a story or a truck in a story, but they're not responding when you show movies. And when you align these and look at the semantic selectivity along this black line, which is the anterior border of visual cortex, you can see that there's a direct correspondence between the semantic selectivity in the vision network and the semantic selectivity anterior to the vision network. And the only case where there is not correspondence is up here at the top of the slide. You see there's two little purple patches here that are areas where people are representing the mental concepts in stories, and it doesn't appear they're representing the mental concepts in movies, but that's because of the way we coded the data. We didn't actually code mental concepts in the movies at all. They don't exist, so we can't find a match there. So I really like this result because I think this is one of the few cases where you see a very striking, clearly non-random functional MRI result that implies that there's probably an underlying anatomical scaffold that should be detectable. It's fairly difficult to find short-range detections right now in MRI because MRI uses diffusion imaging, and diffusion imaging is a very, very noisy medium. It's kind of a prior-dominated science at this point. And diffusion imaging is very bad at small short-range connections. But I think in the future, this is something we want to look at. And I also have a theory that we're going to find these same kinds of relationships, say, around auditory cortex. So part of, auditory cortex is hierarchical, just like the visual cortex. Early auditory cortex represents the sound spectrogram and things very closely correlated with it. Higher order auditory cortex represents the semantic content of the sounds. And I suspect that there's going to be a similar border between higher order auditory cortex and this multimodal system where semantic concepts in the auditory system feed into this more general multimodal conceptual network. If only NIH would give me money to look at that, I could tell you the answer already. All right. So now let's move on to so I've told you about the amodal conceptual network, or the multimodal conceptual network more correctly, and modal conceptual networks. Let's look at long-term memory and executive functions. So I told you earlier that the ATL seems to be a key bottleneck in conceptual representation, likely because it represents the channels between long-term memory and the ongoing active distributed conceptual network. And so we would like to image the ATL to try to understand, what is being represented there and how, and maybe, although this is kind of a fantasy that I don't think we're actually going to be able to solve, trace back where the memory engrams are first like represented in active conceptual networks. The problem with this is that the ATL turns out to be very difficult to image using functional MRI. Orbital frontal cortex and the ATL are basically horrible, and the reason for that is susceptibility artifacts. They're right next to ventricles or air sacs, like the olfactory, air sacs where in your olfactory system, and so the signal to noise is bad. So I'm sorry for the right there. I know what causes that, and I should have fixed it. An MRI physicist who was a former graduate student of mine at UCSF, Joseph Vu, and my postdoc, Matteo, who you saw earlier in an earlier slide, spent about 6 months developing a new multi-echo pulse sequence that dramatically improves the signal to noise in the anterior temporal lobes and in the orbital frontal cortex. You can see actually it's even better, the difference is even bigger in orbital frontal cortex than it is in the anterior temporal lobes. But this dramatically increases the signal to noise that we have available to build models, and has allowed us for the first time to map the ATLs. So I'm just going to skip to here. This is the group model. This is 3 subjects. We actually have, I think, 7 or 8 subjects now that we've done this ATL mapping in. These are the same kind of maps you saw before. The ATL is outlined in white, anterior temporal lobes, and the legend up here at the top left, it just tells you a grouping of, I think, 16 of it's a cluster analysis of 16 kinds of semantic concepts. But remember that we're fitting 1,000 semantic concepts to each individual voxel here. Let's see what else to tell you about this. Pink things are social, green things are visual. So you can see down in the anterior temporal lobes that there's a lot of representation of social and visual information, both at the group level and in the individual subjects. But what's most striking is it seems like there's nothing special about semantic representation in the anterior temporal lobes. It's just a continuation of the semantic representations immediately outside the temporal lobes. It's not like something dramatically different is represented there. It's more a bias. It seems that the anterior temporal lobes seem to represent relatively more social information and relatively less, say, physical information than is represented elsewhere. So you can see here the anterior temporal lobe left hemisphere represents a lot of social and emotional information, and the right hemisphere. And you can see if we look at cortex as a whole, the emotional and social information is much more balanced along with the physical information. So it seems that the anterior temporal lobes are representing more social emotional information. Question?
Yeah, just to make sure I understand how each voxel is being colored, is if it's, say, labeled with the social color, it means that the prediction for social concepts is the best for at that location, and there's tuning for the other concepts?
No, and I'm sorry for skipping over that. So each voxel is a 1,000-long vector, and most voxels are tuned for a bunch of stuff, right? So this is one of these wonderful things that happens in science where you have too much data. So we do principal components analysis. We take the first 3 PCs. We code them red, green, blue. So each color on these maps is some color that represents the loading of these 1,000-dimensional space onto these first 3 PCs. These colors here are just a discretization of the colors. So that the colors on this map are continuous. These are just the discrete categories that conform to different kinds of things. Okay. So again, every one of these voxels is a 1,000-long feature vector, right? That we're clustering. Okay. So I want to mention 2 things about the ATL results. The first is when people end up with ATL damage, one of the severest impairments they have involves social and emotional information. They become very asocial. They can't make emotional judgments of other people. Social information is just completely disrupted, and it's much more severely disrupted than other things. And the second is it's possible that this might have to do with some sort of attentional effect. You may have noticed when we were clicking around in the brains earlier that the social voxels also responded to bad things that happen in families, like confessed, divorced, murder, things like that. And in these data sets, I have to tell you that old newspaper saying of "if it bleeds, it leads" is reflected in these data. People pay attention to violent, nasty, horrible things that happen. That's how Fox News makes money, right? And so I think what may be happening here is that there's just more attention paid to social concepts, and so those end up basically accessing into memory more. They tend to elicit more autobiographical information.
But Jack, differentially activating ATL versus other regions, or you see the attentional effect in the other bits of cortex. There's something about attention to those things that's especially happening in ATL that's not happening in other regions.
Right, and that was what I was trying to say. This is all speculation, of course. But my theory about this is that it has to do with if you see a truck on the street that doesn't involve a lot of memory access. Oh yeah, it's a truck. You have to label it as a truck. You don't really care about the truck. Mm-hmm. But if you see somebody murdering somebody on the street, that you now have to interpret the social context, and it probably starts eliciting a lot of other associations that are in the penumbra of that specific event that cause more memory access, right? All of your current experience is this constant interaction between what's coming in through your sensory systems and long-term memory, but the balance between those two things, the balance between the likelihood and the prior in your Bayesian internal model of the world is constantly changing. Okay, so now the last thing I'll talk about is this executive control issue. How much time do I have? Oh, I'm not doing that bad. Okay, and so I'm gonna, as a stand-in, a proxy for executive control here, you might think I would be talking at this point about cognitive control or about working memory. And instead, I'm gonna talk about attention. Why? Because attention is very easy to measure, and there's a lot of data that shows attention and working memory are basically the same thing. In most cases, most attention experiments, if you relabel the parameters, they become working memory experiments. Most working memory experiments, if you relabel the parameters, they become attention experiments. These two things are very, very similar to one another, and disentangling them is very, very hard. So it's pretty easy to study attention, and I'm a vision guy, so that's what I like to focus on. So I'm going to talk about attention as a kind of a reflection of executive control that's ongoing in these circuits that controls conceptual representation. And I'm just summarizing with this very abstract slide. There's a long line of research, both in primate physiology and in human MRI, that shows that attention changes the tuning of neurons or voxels in a task. And in particular, it seems to change tuning to sort of optimize the brain systems for performing that task. So you can imagine if you want to represent these various semantic concepts here, in a completely neutral world where you have to represent all semantic concepts in this two-dimensional space, you might just tile this space with voxels and neurons that have random tuning peaks, right? But now if I tell you, okay, yeah, there's a bunch of semantic concepts you could see in this movie or in this story, but I just really want you to focus on humans and tell me when a human occurs. Well, now you might decide to perform this task optimally, I'm going to shift the tuning of my neurons or voxels so that they more heavily represent this area around humans. And that'll, of course, be at the cost of everything else. And now if I tell you now pay attention to vehicles, you would shift your attention over to vehicles, which will shift the tuning over this vehicles quadrant at the cost of everything else, because you only have so much hardware to go around. And there's a lot of data that shows this occurs. This is a very phenomenological explanation of what happens, I should just mention, in all fairness to others. There's another computational model that describes this exact same process that comes out of Earl Miller's work in prefrontal cortex, which shows the same effect, and that's by Stefano Fusi. That's this mixed selectivity computational model. This is not a computational model. This is a kind of a phenomenological model. Stefano's computational model is basically a model of how you would implement this phenomenology, and I think there's just not enough data yet to know whether that model is correct or not. But the phenomenology, whatever model we come up with, has to be consistent with this phenomenology. And I want to show you how this works in MRI. Doing an attention experiment like this in stories turns out to be a real pain in the rear end because it's hard to know when people are listening to a story about humans, when they should respond to human. It's very easy in movies. You see a human, you hit the button human, right? But in a story, it's a vaguely story with a narrative arc about humans. When is the human actually, the appropriate response? So rather than do that, I'm going to quickly segue to a completely different experiment. This gave me a way to shove in my navigation experiment, which I'm really excited about. Okay, in this experiment, this is a navigation experiment done by this awesome postdoc, Tianjiao Zhang, who's worked forever on this experiment. He basically built a world in Unreal Engine. This world has about 200 landmarks in it. It's 2 by 3 kilometers on the side. It's got a bunch of buildings. It's got urban areas, suburban areas. It's got roads. It's got trees. And basically, people go in the MRI machine, and they're driving in this world in a car on the road. And they go in the MRI machine. They've got a little magnet-safe steering wheel, little magnet-safe pedals that Tiangel built. They go in the magnet, and they're an Uber driver. So they spend about 10 hours learning this map outside the MRI machine. And then you go in the MRI machine and say, okay, drive to the gas station. They drive to the gas station. Drive to the grocery store. They drive to the grocery store. Okay, that's the experiment. That's all they do. You do it for 2 hours. They go out. They come back the next week. They do it for 2 hours. They get like 8 hours of data. It's an awesome experiment, and data's like coming out of a fire hose in this experiment. It's crazy. It's really cool. I strongly would encourage you to do virtual reality in the MRI machine. It's really, really fun. This came out of some older experiments we did on Counter-Strike. At that time, which is 10 years ago, it was too hard, so this is like the Counter-Strike subspace. But really, I would like to do Grand Theft Auto because... Okay, so this is that experiment. And now what is represented in the brain during driving? Well, all the vision stuff, all the motor stuff, all the navigation stuff. There's dozens of different things that are going to be representing your brain. So Tiangel built 35 feature spaces, and these are all of them. And these were all taken from the motor, the vision, and the navigation literature. So these are mathematical instantiations of various theories about what information might be relevant in this task. Okay? So there's retinotopic information, frame-wise information, high-level navigation information like affordances, spatial semantics, scene semantics, what you attended to, a bunch of motor stuff, motor plans, when you're going to turn next, what your future path is, goal-related stuff, your destination vector, a vector anchored to where you are, a vector anchored to where you started, path integration variables, all this kinds of stuff. Okay. So what we do is we take the movies and the task information, which is your control surfaces, your steering wheel, your pedals, and your eye tracking. We have all that information. Those are all our X variables. We translate them into these 35 feature spaces. This ends up being 25,000 features. And then we do regression. So each one of our 100,000 voxels, we're going to fit a 25,000 dimensional feature vector to that voxel. And if you want to know why I advocate doing encoding models rather than like the older methods for like MRI, like SPM, imagine trying to do this in SPM. It would be like impossible, right? Encoding models, no problem. This is an easy problem to solve. Well, not easy. Easy in professor time, hard for the graduate student. Very easy for me. All right. So this is, after we did this, we fit all these models. This isn't about navigation. We did model connectivity. And so basically on the horizontal axis here is the 35 models, basically oriented from vision to motor to navigation. And on the vertical axis here is the clusters. And to make a very long story short, there's a bunch of motor clusters, there's a bunch of vision clusters, and there's 3 unique navigation networks that appear to represent motor bias navigation information, vision bias navigation information, and abstract navigation information, and these are distributed in different places in the cortex. But that's a talk for another time. What I want to focus on now is attention. So when people are driving in this MRI experiment, they're looking around the screen all the time, and they're looking at different things in the world. I'll look at the traffic sign, I'll look at the street sign, I'll look at the car in front of me, I'll look at the building, and we're tracking their eyes, and we have ground truth from the game. So we know exactly what category of thing they're looking at, right? So we can look at what happens to these representations of this semantic stuff in this task. And in this condition, they're passively viewing random video clips. This is not actually from the driving data, but you could make this from the driving data. And we have, and you basically get the same thing. And if you pull out all of these different semantic categories of objects, you get a map that looks kind of like this. So these red areas are representing vehicles, and you can see like the hippocampal place area, the region in front of the RSC, and the occipital place area, which are all kind of navigation-related areas. They're representing vehicles and pedestrians and stuff like that. Roads are represented, different places, all kinds of stuff. Anyway, okay, but this is passive. People aren't really driving here. What happens if you put them in a driving experiment? Everything changes. So now you notice what is represented all over the visual system now. Traffic signs, poles, roads, stuff that matters, vehicles, and a bit pedestrians. Stuff that matters that's relevant for the task is represented here. I didn't mention there's other cars and there's pedestrians in this environment, right? So the map completely changes depending on your task. This is essentially the same input, different task, and everything changes. Why? Because tuning of the voxels is shifting toward the attended category, and tuning is changing everywhere. Now at this point in the talk, everybody's going, what in the hell is going on? Why does this work? So I'm going to give you a really simple intuition for this. You all know like convolutional neural networks like AlexNet or VGG, right? Think about when those networks are being trained. Initially, all the weights are random. You apply a cost function. You backprop the gradients that you need to adjust to optimize for that cost function. And what happens is the tuning of the individual units in that neural network change. All the time during learning, when you're building a deep neural network, the tuning of the individual units is changing. And units at the low end, in the periphery, analogous to the periphery in vision, their weights change very, very little, and the representation doesn't change much. But neurons, artificial neurons in this case, at the top, their tuning is completely changing depending on the task. And what is the difference between early versus late neurons in this hierarchy? The connectivity they have, their potential connectivity to everything else. So I think all we're seeing here is that in humans, unlike artificial neural networks where you train and then deploy, humans are constantly being trained and constantly being retrained. And these dynamic attention networks are constantly updating the weights, and that's changing the representation at all levels of the network. Oh, this is just the difference. Okay. The biggest attention effect we've ever seen is what happens with representation of vehicles. So this is the representation of vehicles if you are in the passive viewing condition. Red is more vehicle representation, blue is less vehicle representation. This is in the active condition. Now you might say, what are these vehicle representations here? These turn out to be all now vehicles are being represented in the animate related areas. Where you like body related areas. Basically when you're passively driving, a vehicle is a mechanical thing that you don't care about. When you're actively driving, now a vehicle is an animate agent to you because you have to exercise theory of mind because you have to figure out what that other vehicle is going to do. So the representation of vehicles completely changes from one condition to another. As you can see, these are basically non-overlapping. Right? It's a 100% shift. Yes?
Is the tuning changing biologically and that's what enables a person to do the task? Or is the tuning change just something that you sort of experimentally measure that's a side effect of something else changing in the brain?
I'm not exactly sure what that would be. We're measuring changes in blood flow here and blood oxygenation. So presumably these tuning changes reflect changes in the underlying metabolism. Now the thing to remember about MRI is, everything's rectified. So it could change inhibition or excitation because both of those are going to change blood flow, right? And there are a bunch of other weird MRI artifacts. In MRI, most of the data in the standard pulse sequences people use in MRI come from blood vessels that are about the size of your voxel. So this would be coming from blood vessels a couple millimeters across that are pretty far from the columns that are actually doing the computation. They may be displaced by a couple millimeters. But other than that...
More conceptually, does a tuning change have to happen in the neurons in order for the people to do the task? Or...
Oh, no. The tuning has to change in the neurons for people to do the task well, right? So I think you could do the task badly just with the base representation, but what you're trying to do is optimize for the task. So if you go home tonight and your little cat Fluffy is missing, your brain tries to turn into a giant Fluffy detector. Where's Fluffy? Where did Fluffy go, right? You start thinking about generating all this Fluffy related information about where she might hide and where you found her in the past. And your visual system's not going to change that much, but your prefrontal cortex is now a giant Fluffy detector, right? And weirdly, interestingly, I should mention this from our 2013 paper, there's one brain network that does not shift toward the attended category. That turns out to be the default network. So for those of you who are in the MRI world, you've heard about this default network. It's the network that becomes activated when people are lying in the MRI machine and told not to do anything. There's no such thing for humans unless they're Zen masters and not doing anything. What they do in this task is they start ruminating. Boy, this magnet is noisy. I'm very uncomfortable. When do I get my $25? I'm hungry. They ruminate. And the default network is very highly overlapping the internally directed attention network and the rumination network. Okay? So I think what's happening in this, and there's some other, believe it or not, mouse data that's consistent with this, is when you're in these tasks, a lot of what your cognition involves is information, say in the Fluffy detection task, it's information about Fluffy that's not directly related to seeing Fluffy in the environment, right? So you're internally generating I remember all the beautiful times I had with Fluffy. Usually I get home at this time and I start petting her and she runs up to me and she's not here today. Oh, maybe she's hungry, right? Maybe she got out the cat door. And you don't want to respond to those. You don't want to say, oh wait, there's fluffy, she's in my hand. Okay, everything's fine, right? So you detune those parts of the brain. Don't respond to any fluffy related signals coming internally. Only respond to fluffy related signals in the external world. Attention is cool. This is a way more interesting thing with attention than like most people's studies of attention, which are like gain and baseline shifts and noise decorrelation. This is really representation is cool. Okay, so I just want to mention there's a lot of brain research. Sorry, there was a question in the back. I'm sorry, I didn't mean to ignore you.
Yeah, kind of related to how profound and how long-lasting and how physical these changes are. What happens if you expose the person to passive viewing, you do your mapping, then they have a more active driving experience, and then you show them the exact same passive viewing again and do your mapping. Does that look
we did that, and then they just go back to the default, the passive state. Yeah.
So you don't have any lasting changes from the...
Well, there's a key word you said in there, any lasting changes. It's not obvious if you just look at the maps. I can tell you that. However, this brings up a really interesting point, which is, I used to say, and until about 6 months ago, I would have argued, attention is just short-term learning. Like, how do you learn? You pay attention to something, and you keep paying attention to it, and eventually you learn it. And, the nice just so story would be those are kind of the same circuits. But there's some mouse optogenetics data that has come out in the last 6 months. My name lesion is affecting me. I can't remember whose lab it came from, sadly, where it suggests that the attention circuits and the learning circuits are kind of two different things. But that doesn't mean that attention doesn't influence learning. I strongly believe that it does. So I think in the long term, if we had a more subtle way to look at these data, we would see that. What time is it? Oh, I'm over time. Okay, well, I didn't even get to the part I wanted. I should mention, since Danny did this cool dog imaging experiment, there's a question of, do dogs have multimodal representations? I suspect it's true, they probably do, but they don't have language, so they're going to be kind of less interesting. But dogs, all animals need to integrate information, right? I don't think a dog has a visual sense of the world and an auditory sense of the world that are decoupled. They're combined together in working memory circuits. Okay, I was going to talk about individual differences, but I'm not going to talk about that because I didn't get to it. But I do want to just give a shout out. Most of the MRI data that is out there is the group level, and we do have a new method for looking at individual differences in groups, and that basically uses this optimal transport method. So if you think about a brain area as being a bunch of voxels tuned in a high-dimensional space, then or a whole brain being a bunch of voxels tuned in a high-dimensional space, then you can look at the difference between the tuning in this high-dimensional space, the distribution for an individual versus the group. And there's a theorem called optimal transport that actually tells you if you want to align an individual distribution to a group distribution, precisely how you have to move each individual point. And since in this cloud these points are voxels, this tells us exactly how the tuning of each individual voxel differs from the tuning of the group. And that's to make a very long story short, because I'm just going to skip, oops, well, I went a bit too far there. So this is the individual semantic map of one person. And this isn't the semantic map of this person. This is the differences in semantic tuning of this person from the group as a whole. So when this person listens to stories, they respond, their brain responds and represents more, I should say. Their brain overrepresents arresting, officers, chaplain, family, policeman refused. And it overrepresents solid lifting, paid, grinding, slide, hook. And it basically just represents at the group level these concepts up here, which are certain such extent specificity specific. Now, I'm going to tell you two things about this. A, don't try to interpret this. Who the hell knows what's going on? This is probably some combination of attention and learning. Everybody thinks different things in these stories. But the reason I think this is really awesome is if we ever want to take fMRI and use it for the clinic, which it is not used for now, we need to be able to do semantic mapping in individuals, and we need to identify individual differences very, very quickly. And this provides a way to do this. We can now semantically map each individual, and we can tell how that individual differs from the group. And that means we could also tell how that individual differs from, say, the neurotypical group versus, say, an autism spectrum disorder group. and see which one of those groups they're more likely to be in. And that's kind of the future of this direction of research. Anyway, I'm sorry I talked too long. Thanks very much for your time. Question? You know,
more data-driven. Sorry, I'm Harry. I'm a fifth year.
Yeah, hi, Harry. You know, there's nothing wrong with that. I think basically functional, you know, what I told you about functional connectivity is not like the people in functional connectivity are unaware that this is a problem. They're fully aware that it's a problem. They just want to avoid it as much as possible and fix their data as much as possible. So what usually happens in functional connectivity is most of that data is collected in resting state because it's easy. And so the networks have to be validated with some other task. So usually there will be some task-specific network that's recovered using functional connectivity, like maybe in a vision task, and then that network will be used as the canonical network to just map some unknown data, right? There's nothing wrong with that, but you know, as a first cut. But the problem is, again, I always encourage graduate students to pay attention to the difference between the noise floor and the noise ceiling, right? The noise floor is statistical significance. It's like, do I have a result that's not random? And scientists are, you know, selfish, self-centered people just like all other humans. And so scientists kind of pretend that that's all we need to do science because it's the easy thing to do. My data's not random, everything's great. But that's not actually what we're trying to do in science. Non-randomness is necessary but not sufficient. What we want is a model that explains all of the variance and that generalizes outside the experimental conditions. And that's the noise ceiling. And we always want to be going to the noise ceiling. And the only way to calculate the noise ceiling is to actually calculate that thing, which is a calculatable quantity, and to do predictions relative to the noise ceiling. And I think if you're doing anything like just using the group model, well, that's a different issue. If you're doing things like using these canonical networks, it's usually very difficult to figure out what the noise ceiling is and how close you are to it. So you know that you have a result that's not random, but you don't know if it's good, right? A significant result and a good result, a meaningful result, an important result are not necessarily related, other than the fact that any important result has to be significant. So I'm a very strong advocate of focusing on prediction accuracy and effect size out of set. And I'm also a big advocate of using separate fit and test data sets. There's no journal in neuroscience or biology or psychology that I know of that requires separating fit and test data sets. And you guys all know that there's a giant problem with type 1 error in psychology that unfortunately has also infected biology, since most biologists learn their statistics from a psychologist. And so this problem is a non-problem, right? The psychology community, for reasons I do not understand, is trying to come up with all these arcane ways to solve the replicability crisis, for example, pre-registration. And you know, my view is you cannot solve a crisis with more paperwork. That has never worked in the history of the world. Can we solve this in a way that has less paperwork? Yes, we follow the modern method of data science. So I'm going to tell this really quick story just because I love it. If you want me to shut up, just let me know. I'm a professor, I could talk for 8 hours. In the 1990s, artificial intelligence was almost dead. They'd been lying to the military for 50 years, telling them that general artificial intelligence was just around the corner. And the military figured out, this is useless, and they weren't going to pay them anymore. And there were all these meetings in the machine learning AI community in the 90s saying, what do we do? How do we fix this? And they realized something really, really important, which is science is data with politics. It's data because there's the data out there, and it's politics because there's humans involved. So it's data with politics. And the problem with AI is there's no data. So now it's just politics. And that's a really bad for a field, right? So there's no ground truth that anybody had to obey. So they came up with these very strict, principled methods, which grew into modern data science. So having contests where some of the data is hidden from you so you can't overfit, separating fit and test data sets, separating your fit set into the regularization set and the true fit set, which is called, I think, training and validation in machine learning. All of that stuff came out of AI. And all of that stuff solves these problems. Nobody needs to do pre-registration. If you just follow good methods of data science, you're not going to have to worry about type 1 error, and you're going to be generalizing because you're going to be testing your test data set under conditions separate from your fit data set. So that's kind of where I'm trying to push the field. What I have noticed is, I like doing development. Most people don't have time. They've got more important things to do. I don't actually have any theory about how the brain works, so I don't mind doing development because I don't have a theory to test. Most people have a theory, and they want to test a theory, and that's fine. And so they don't want to do development. So we're trying to make the software available to people so that they don't have to do it themselves. And so everything I talked about today, the brain viewers, that's PyCortex. It's publicly available and online. The GLM fitting stuff, all the box-wise encoding models, that's our Himalaya software package, all available online. And there's tutorials for how you use Himalaya that are really great tutorials. Anybody can learn to do this. And then this model connectivity, the code is being refactored so that other humans can look at it. And after that's done in a month or two, that will also be online. So all these tools are available for anybody to use. The fundamental rule of graduate school, remember, is if your advisor's doing it, it's the wrong thing. So if you're a grad student, your advisor will be annoyed that you're doing something they don't understand, but in the world, that's how science evolves. So just go ahead and do it.
You said dogs don't have language, which makes the data less interesting. Have you done any
Well, that's a guess.
Yeah. Have you done any studies with language area lesions or English native speakers and non-English native speakers?
We did. We actually, one of my postdocs, former postdocs, Fatma Deniz, who's now a professor at the Technical University of Berlin, did a big bilingualism study with Chinese, English bilinguals. And these were all, just to make it clear, they were Chinese graduate students studying at Berkeley. So their English facility was generally pretty good. They could survive graduate school, right? And we had them listen to stories, did full semantic mapping in Chinese and English. They actually, I think they read the stories, they read them in Chinese, they read them in English. It was all counterbalanced, all of that. And the semantic maps, I gotta tell you, look remarkably similar. So this obviously disappointed Fatma. She'd spent years and years and years on this. Like her and 5 other people in my lab was a huge amount of money and time, people's effort. So we've gone in, retrospectively tried to find out what is in here. Generally, bilinguals seem to be representing semantic concepts in the same regions, but there are subtle differences. And 3 of them I can mention are, it seems like numbers are represented differently in the 2 languages. And for those of you who are bilingual, you may notice it's pretty common that you think about numbers in your native language, or you have a tendency to. And we think that's why. So numbers, and this number network of all the networks in the brain is like the most special kind of isolated, cognitively impenetrable thing relative to all the other networks. So that kind of makes sense. It may be that social concepts are represented differently in the 2 languages. We're not quite sure yet. And the other thing that's interesting is there's an old theory out there. This is just brand new. So in a week, this may change. I'm just telling you what the latest thing is. There's an old theory in bilingualism that concrete concepts will be the same between the languages, but abstract concepts will be different. And in these data, it looks like very concrete concepts are the same, and very, very abstract concepts like truth are the same, but it's the concepts in the middle that are different. So that could be an artifact, I'm not exactly sure, but there are a few things like that. But the differences that we find are going to be subtle.
What's
They're not going to be
in the middle?
Like something like friend or something. It's kind of half abstract, right? But like truth or something like that would be. That's obviously an abstract concept, but it means the same thing to everybody.
Okay, given the time, we'll take one more question, and then you can speak with him in the reception.
Or you can email me.
Oh, so how do the representations you get when you're retrieving experiences from long-term memory compared to the representations you get when you're actually experiencing, like those movies, for example?
So nobody has done that experiment directly, and it has the same problem that I mentioned with respect to movies and stories, right? That if we show a movie and show a story and tell a story of that movie, they're not exactly equivalent, and so there are attention differences between the 2 cases. We have the same problem if we ask somebody to remember versus just watching something. So we have done imagery experiments where people watch a movie and then they have to retell the movie, or they listen to a story and have to retell the story. And there's also been several visual imagery experiments where people see a visual image and then have to reconstruct the image in their mind's eye. So in the early visual system, for like imagery experiments, these look to be very, very similar. But as you go into the spooky bits of the brain, more anteriorly in this multimodal network, it's just completely unclear at this point. Yeah. Science is hard. Thanks for coming.